Hmm. All right. All right, all of you who are joining us on Zoom and Facebook, we've just got about 1 minute and we will get started. All right, looks like we have turned to 4 o'clock. Welcome everyone to Crow Canyon's Thursday webinar series. Thank you all for joining us. Before I introduce our distinguished speaker today, a couple of our usual introductions. We like to start with our land acknowledgement. Crow Canyon acknowledges the Pueblo, Ute, Paiute, Dene, and Jicarilla Apache people on whose traditional homelands our campus sits. Our work is not possible without Indigenous people in the past, present, and future, and we respectfully recognize and honor ancestral and descendant Indigenous communities for their contributions to all humankind. We are grateful to all Indigenous people and support the preservation and protection of cultural traditions, ancestral connections, and sacred lands. Thank you to all of our Indigenous partners today and over the years. I usually open the slide with, if Zoom is giving you headaches. Zoom is always giving me headaches, and particularly the last couple of webinars, but hopefully things will go well today. You can put your questions in the Q&A, and Jonathan told me earlier that he would actually watch the chat, so you have special permission today to put your questions in the chat so that he can grab them real time if you like. If Zoom is truly driving you up a wall, we are live streaming on Facebook, and this lecture will be on our YouTube channel, along with all of our other ones. If you haven't signed up for our YouTube channel, please go ahead and do so. We have some really interesting webinars coming up over the next couple of weeks. We have a hidden history, an overview of African American history in Northern Arizona with Margaret Hangen next week. And the week after that, we have our friend Dr. John Douglas, always does an amazing talk, Abandonment Issues, the Indigenous People of the Casas Grandes Valleys and the Legacy of Pacime. So both really cool webinars. So please tune in the next couple of weeks as well. As always, this entirely free webinar series is all because of you all who donate. Almost everything we do at Crow Canyon is based on donations and philanthropy. Thank you so much to all of you who are always giving to these webinar series and to the rest of Crow Canyon's programs. We are so grateful for you, and we truly would not be here without you. And with that in mind, I have a special pitch to make today. We are a nonprofit, entirely dependent on everyone's generosity. And you might have seen that we have had an appeal going since the spring that we call Dive into Decades of Data. And we had a $50,000 goal for this appeal, and we're so close. Jonathan, this is a near and dear to him. And so he said he would say a couple words about why this is so important to us and what your generosity would be used for.
Yeah, absolutely. So before I begin, just want to say, if you're wanting to contribute to this, to support us for this, we just dropped the link in the chat. You can also use the QR code here. But essentially what we're trying to do right now, I don't know how many people know about Crow Canyon's database, but we're talking about archaeological data that was recorded, analyzed in roughly the same way for over 40 years. So it truly is one of the most comprehensive archaeological databases in North America. I do not think it is an understatement to say that, or an overstatement rather, to say that. But one of the things that we're trying to do with this appeal is make sure that other scholars and maybe even students that want to learn data analysis techniques can actually access our data in kind of up-to-the-minute ways. So that's what this whole thing is about. It's about updating the front end of our database so users can use it better. So if you could help us out, we would really appreciate it.
Thank you, Jonathan, and thank all of you so much. I mean, so much generosity to have even gotten us this far, and we're just really hoping to be able to get that needle all the way to the finish line. So thank you, Jonathan, for explaining, and thank all of you for your generosity and for your consideration of helping us get over the finish line for this. Today's webinar is a special one, the Four Corners Lecture Series, which pre-pandemic was always in person, and then we partnered with them to be able to offer it in the webinar series. And we really value the Four Corners Lecture Series in general and our partnership and being able to provide them. And we're so grateful they ask us to participate in the lecture series and our Crow Canyon scholars such as John. So it includes 12 local organizations who have a shared mission with Crow Canyon about protection and preservation of cultural and historical resources. So really excited to participate with all of our friends and partners in this lecture series. Without further ado, I am so happy to introduce my friend, my colleague, my neighbor, Dr. Jonathan Dombrosky, who I know so well that I failed to call up his bio and have that in front of me. So I am a little moderately embarrassed.
Well,
Why don't you introduce yourself, Jonathan?
Yeah, it's not a big deal.
We're actually really good friends and colleagues.
Yeah.
So Jonathan, tell the folks at home a little bit about yourself.
Sure. So I'm the environmental archaeologist here at Crow Canyon. I got my undergrad in anthropology at the University of North Texas. I got a master's degree in applied geography with a focus in environmental archaeology at the University of North Texas. And I got a PhD in anthropology at the University of New Mexico. I did my postdoc here at Crow Canyon. And then I recently, last year, at the beginning of this year, started this environmental archaeologist position. And I'll introduce all the kinds of different topics that I'm interested in. You'll see all of it, and you'll hear it here in this talk. So let me share this. Share my screen. Can everyone see that? Is that coming through?
Perfect.
Great. And you can see this little laser pointer.
Moving.
Yeah, that's very fancy.
Yes.
So I just wanted to say to everyone, actually at the outset here, as you can kind of see with this, that there are images of animal skeletal remains. If that is not your thing, then I totally understand. You can log out. I kind of kept them few in this talk, so there's a lot of illustrations, but there are a few times where I do point them out. Another thing here that I just want to say is I'm going to do this in a little bit of a different way than usual. I'm going to try to make this not so one-sided. I was going to try and keep an eye on the chat, or the Q&A rather. But it's pretty hard to do that. I'm asking if maybe Liz could do that. I want to keep the presentation pretty conversational. If there are any questions that come up, we can answer them on the spot. One of the things that I do is I get super chatty, get really excited about stuff, and I'll just like not explain something really major. So if something like that happens, I want to create room for people to kind of, ask about that, and I can slow down and talk about it. So what I've done here is I've just packed in information from the last 2 years that we won't be able to cover. So I'll just at probably 4:50 my time, I'll stop. We can have some extra questions and time for that. But I'll just kind of go. So basically, the whole goal of this talk is to talk about the last 2 years or so of zooarchaeological research from the Northern Chaco Outliers Project. This is actually kind of a wrong title. I would say the more accurate title right now would be 2.8 years of research, of zooarchaeological research. We just finished our kind of third major season of analyzing things, but we're in the process of reporting and working on the annual faunal report. So I'll talk potentially a little bit about that research from the previous zooarchaeology intern. Her name is Chelsea Betts. She was excellent zooarchaeology intern. She did awesome work, and I definitely hope I get some time to talk about that. So From the get-go, though, I just want to make sure we're all on the same page. I'm going to start really, really general. I know some of you already know this, but maybe some of you don't. So if I'm talking about these general kind of time periods, here in the Southwest, we call that the Pecos chronology. But today, we usually, especially here in the Mesa Verde, Central Mesa Verde region, we talk about Basket Maker 3, the kind of start of these pit house villages, the start of above ground kind of villages, full-on village life around Pueblo 1, and then agricultural intensification, larger villages around Pueblo 2, and the last bit of, pre-Hispanic occupation in this area around Pueblo 3. Today's talk regarding the Northern Chaco Outliers Project is about this Pueblo 2 time period. This is essentially from 8900 to 1150, and the time period, if anyone has heard of Chaco Canyon, one of the most important archaeological sites in North America, located south of us here in New Mexico, the Pueblo 2 period around 1050, they refer to this, Steve Lexon refers to this as the time when Chaco goes nuts, right? So 1050, it reaches its height. And one of the really super interesting things during this time is that the height of Chaco's power pretty much extends to a region the size of Indiana, to the state of Indiana today. And how this is measured is through these things called great houses. Great houses are multi-story buildings built in kind of a very similar architectural style. There's arguments about this stuff, so I'm glossing over a lot here. There are kivas sunk into these buildings. There's kind of a specific material culture, some certain specific artifacts that are found at these sites that tie them back to Chaco. So we can kind of measure that. So it's this huge kind of sphere of interaction of the Chaco system, as it's called. So these are what Chaco outliers are. The Northern Chaco Outliers Project is about a basically cluster of great houses. So here, the Northern Chaco Outliers Project area, I've pinned it in Google Maps, if people prefer to see it like this. This is where we're talking about. This is the Four Corners region right here. Four Corners is here. So this is where I am right now in Cortez, and this is where the NCOP study area is. And so the NCOP study area, really briefly, I'll show you some maps here in a little bit that kind of show you what this location looks like. But just really briefly, to get you situated, it is a cluster of 4 great houses. And this kind of cluster is really rare in this huge Chaco system. I think the quote, the statistic is that of all of these outlier sites, these Chaco outlier sites, less than 5% of them have 2 great houses, and less than 1% of them have 3 or more. Well, here in the Northern Chaco Outliers Project, we've got a cluster of 4 great houses, and we call this the Lakeview Community. So I'm going to be throwing around all these terms: Northern Chaco Outliers Project, the Haynie site, which is part of the Lakeview Community, but the Lakeview Community, and I'll show you pictures of it, has 4 great houses. So this project is all about how does a cluster of great houses like this interact during this time period? How do they relate to Chaco? How do they maybe form their own identity? Things like that. So this is generally where we're talking about. To get a little more specific, the Northern Chaco Outliers Project has 4 research domains. They are briefly just talking about inequality, public architecture, identity, and then human-environment interactions. So when we talk about social inequality, kind of what we're talking about is measuring access to different resources potentially within this community. Does, say, one of the great houses have access to something different? Are there specific signatures related to that? Could it be related to ritual knowledge? Things like that. To understand public architecture, what we're talking about are things like great kivas. How are people using great kivas in public architecture within the Lakeview community? How are they using them at the Haney site, which is where I'm going to focus at today? And then also, how does public architecture use relate to Wallace and Ida Jean, which are the 2 other great houses, these sites? And I'll show you pictures of them. And also kind of related to this idea of public architecture is identity, right? So identity formation. So how do people create public architecture? How does that happen? What is the knowledge that's passed down to do that? And you can use this as kind of a fingerprint for different groups of people and knowledge bases kind of on the landscape, which is really fascinating. And we call it the communities of practice. Again, comparing this kind of identity formation and communities of practice to Chaco and to the other sites in the Lakeview group. And then the other topic that's really near and dear to my heart, but also applies to all these other research domains too, is just generally understanding human environmental relationships better. So how did people, did they struggle with drought? Did they struggle with resource depletion? Or actually, how did, kind of flipping those kind of really negative things on the head there, how are people sustainable through time? I'm really interested in that kind of question, sustainability through time, and people making these active decisions for sustainability. So again, these are just the four research domains, general research domains of the Northern Chaco Outliers Project. And I would be remiss without saying the person who came up with this all these research domains. Her name is Dr. Susan Ryan. She's actually probably right over there right now across the way in her office. So she designed this project, and then the zooarchaeology is just one part of this. Many people are working on this project. And if you want to know more, there's all kinds of reports on our website that you can look at. Okay. If there's not any pressing questions of all of that I've just flown through, we'll kind of give you a little tour of the Haynie site of this general region, and then we'll get to the zooarchaeology, which I hope is what a lot of you are here for. So this is kind of the classic Crow Canyon map. Right here is what we like to call the Central Mesa Verde region. And again, this is just the same kind of map that I was showing you before. This is just where the Lakeview community is, smack dab here. This is Mesa Verde National Park. You can see all of that. Cortez is right there. So we're going to zoom in even further. So this is the Lakeview community, a cluster of 4 great houses, Ida Jean, Haney, the Haney site complex, which includes 2 of the great houses. We call them the West Great House and the East Great House. You'll get pictures of them soon. And then Wallace, right here to the south. So that's the Lakeview community, a cluster of 4 great houses that are located within about 1 kilometer of each other. And there's other small habitation sites around here. So again, to reiterate, the Northern Chaco Outliers Project is all about how these sites relate to each other, how they make they're working together or not, right, through time, and how that relates to Chaco and the Chaco regional system. Okay, so let's go in even further here. So this is the Haynie site right now. So we've been working here. Field work was initiated at the Haynie site, I believe, in 2017. We started in mapping in 2016, but in 2017, this is the property line. And what you can see is the remnants of the West Great House are right here in the backyard of this house located here. This back wall is all that's intact right now. So this is the West Great House. And then we have to the East Great House, and the property line runs right through the East Great House. So we've been working mostly over here. I'll show you some, you can see all these units that have been open. That's what these pictures are here. We've been working over here and most recently, but we're trying to get over here in the next few years to finish up the Northern Chaco Outliers Project. Also, there is a auto body shop that was on this property that the previous owner, Ralph Haney, he worked and had an auto body shop. So that's the site. This is the Haney site. Just to give you an idea of what the West Great House used to look like. Right now, again, I just said that this back wall is all that's intact now, but this is what it used to look like. There was mapping done before this fell down. There's a kiva sunk into the Great House, kind of like a half moon kiva there too. This is what it looks like today. This is some photogrammetry work that was done, mostly by Grant Coffey, to show what this basically looks like, this back wall of the West Great House. The East Great House, a little bit different, less rooms, more kivas, and that's what the floor plan of it generally looks like. that's what it looks like today. The property line again runs through right here. So that's what we're talking about here in the Haney site. So the zooarchaeology that I'm going to discuss is all about from these kinds of contexts. But actually, one of the things that happened while we were doing this, so these are most of the units that have been opened up from the excavations that we've done. And what has happened actually is that we have realized that there is a large basically P1 component to this site and a room block here, an arcing room block, as well as other pit structures and other habitation, smaller rooms and other kind of arcing room blocks too. So we've gotten really, really concentrated, given the material that we've recovered, really concentrated on how does a cluster of this great house community cluster develop through time. So most of the fauna that I'm going to be, if not all of the fauna that I'm going to be talking about today, comes from these earlier contexts, not necessarily from these later, right, like firmly associated with the great houses, the East and West Great House, earlier components. But we're going to do a general overview of those things. Okay, Liz, do we have any questions thus far?
Someone just popped in right as you said that. Yes, how common are half moon kivas?
I have no idea. I have no idea, but I know someone who will know, which is Dr. Susan Ryan, who just nerds out about architecture. So if you want to send me an
I'll
email,
text her.
okay, we can definitely keep that going. And if she's in, she said she might be able to pop in. If she's in, please, Susan, answer. Okay. Now we're going to talk about zooarchaeology, which I promise I have more content knowledge about than architecture. I think. Okay. So I just kind of want to get people to understand the general zooarchaeological process as I think about it. Again, don't want to be dogmatic here. This is how I think about it. I would not walk away from this talk thinking that this is how all zooarchaeologists work. But for me, I think about it kind of in four parts, which is you kind of need to know what you're going to be talking about. You need to think first, right? What are the topics that you're interested in generally? You don't need to decide on anything before you're looking at stuff, but generally, what are you going to be thinking about? Because it informs how you look at things. Then you actually identify fauna, actually faunal remains. And when I say that, I mean non-human animal bone recovered from archaeological sites. So you actually have to go through and identify that. That's one of the skills that I have based my life on. Then once you have identified all these things, you have to analyze that data, which is an arduous process. Anyone that has worked with me can attest to this. If Chelsea's in the group right now, she knows. And then you must report it in an open way. So to go through this again, you need to kind of think, right? And for me, I'm really interested in questions about resource choice. How are people making decisions about food? And how are they making decisions about food, especially in the face of a changing climate? What are environmental conditions like? And another one that's really near and dear to my heart, what is preservation like? And how do we know what we think we know? We do not get an intact record when we're dealing with archaeological material, right? So really handling that in a very thoughtful way is important to me. You'll also hear this called the study of taphonomy among zooarchaeologists. But essentially, it just means accounting for preservation or an incomplete picture. So then you must identify, and we identify things like the type of bone, the type of animal, preservation signatures, these taphonomic signatures. So how was the bone broken? Was it burned? Was it eroded? How was it? Was it burned not black, but burned so hot that it burned white? All these kinds of things we record. And then once we're done recording, we analyze the data. The typical things that zooarchaeologists are talking about are things like animal abundance, how many animals were actually on the landscape, or like trying to maybe reconstruct that. It's super hard to do. What was on the menu? How much of this thing was on the menu for people? I also like to analyze sampling effects. One of the things that I really think a lot about is how are we biasing the record that we are looking at, like through screening and things like that. And again, analyzing preservation and taphonomy. And then again, you got to report things. That's part of the process. If you're doing all of that work right there and you're not reporting things or writing about it and sharing in an open way, then what are you doing? is my kind of opinion on that. And so we'll talk a little bit more about that too. If anyone is not familiar with me or my work or anything that I've done, one of the things that I've really kind of focused most on is fishing in the Middle Rio Grande in the Pueblo IV period, so around 1300 to 1600. And I kind of answered questions about fish as food and how environment could be playing into decisions to go fishing or not. So I am a sucker for the fish. These are suckers as well. One of my favorite fish, the smallmouth buffalo, is what this is. And we have fish at the Haney site as well. I won't really be talking about them much, but they are there. I also am really interested in this subsistence practice called garden hunting. So really how people draw animals into garden plots to then create protein for themselves. Really interesting process. I've worked on that quite a lot. I'm working on a project right now where I'm actually trying to take the stable isotope values, bone chemistry values, essentially, of individual Cooper's hawks to see if we can actually, in the archaeological record, predict individual animals, if that is doable or not. So that's also one of the things, as well as the Haney Fauna, that I'm working on. So that's just a little about me and a little bit about what I think about on the day-to-day basis. This is not what all zooarchaeologists think about. This is just me. I just want to reiterate that. I'm biased in the things that I'm interested in, and I like to be upfront about that. So let's talk about the Haney fauna. Let's get to that. At least I've got a bunch of different topics that I can talk about. One of the things that we'll definitely get to, I'm hoping to get to these three things. We'll talk about basic bone counts from Haney thus far. Again, things are not complete at Haney. So I just want to reiterate that we're still working on the Haney fauna. It is not a done deal. Again, not a done deal. So we're going to talk about basic counts as they exist right now to kind of just talk about how does Haney compare to other sites that especially Crow Canyon has looked at. Then there's two taxa, general taxonomic groups, animal groups, that are really interesting to talk about from Haney. One is a bison remains that were recovered from the site. So we'll give that some time. And then another one is arcanids recovered from the site, particularly a wolf specimen. That is very interesting. So though we're definitely going to get there. I bet you that's all we're going to get to. But there's a ton of other things that I've looked at. And if you're curious about any of those things, then you can go online and see our reports. I'll also have a link to the open access reports that you can kind of see what we've been working on year to year. And again, to reiterate, we are working on the third one as we speak. So there's more to come. Okay. So, the identify part of that zooarchaeological process, I just want to kind of give an idea of what we're talking about when zooarchaeologists are sitting down and doing what's called faunal analysis. And so this is actually a pile of fish bones, again, a topic very near and dear to my heart, but this is still what we're dealing with. We're dealing with bone fragments. And so we're trying to figure out what bone it is first. That's really important. If you don't know what bone it is, then you can't go any further. What bone it is first, and then what animal that could belong to through a series of comparisons with a library of skeletal material. So this is a really big task. And so the Haney fauna is also very large, and I need help to do that. And luckily, I have had the pleasure of working with 3 interns over the years that have helped me with faunal analysis. Eric in 2022, Anna in 2023, and then Chelsea Betts most recently. And so they've helped. Right now, in terms of what we have analyzed, I estimate that given what's in the lab for the Haney site, we have only analyzed a little bit more than a quarter of the fauna that we have in the lab right now. And that quarter comes up to about over 8,000 specimens, which is a very large sample, okay? 8,000 specimens is pretty, and that's how it stands right now after working with Chelsea and us IDing over this summer. 8,000 specimens. Basically, what I'm trying to communicate is the Haney fauna is on track to be one of the largest, if not the largest, faunal assemblage that Crow Canyon has ever analyzed. It's a very, very big faunal assemblage. So it's going to take time, but we can still learn things while we're going through this process, which is what this talk is all about. So let's talk about some of the basics. So here's one of my kind of favorite graphs. It is just a laundry list of different, you're going to see a lot of graphs here coming up, which I love. This laundry list of taxonomic names, common names for animals here on the y-axis. And then on the x-axis is a value called percent NISP, or the number of identified specimens. And you're going to see this again. And all that is very simple, is a tally of the number of fragments or whole bones that I've looked at from that taxonomic group, from that animal group. So the way to interpret this is the diversity of things that we're looking at the Haynie site so far, and this is their abundance, and they are ordered by abundance. So the most abundant thing that we have down here are cottontails, right? They are just under, I would probably say, 18% of the faunal assemblage that we've looked at so far. The raw NISP count is right here next to each bar. And then as we go, small mammals, medium artiodactyls. Artiodactyls are things like deer. Medium artiodactyls are things like mule deer, pronghorn, or bighorn sheep, usually mule deer. Medium mammals, jackrabbits or hares, rabbits and hares. Large birds, dogs, wolves, or coyotes, canids are what we call those. And this is really typical, okay, of the kind of typical faunal assemblages that we see really in the Central Mesa Verde region and the Southwest in general. These faunal assemblages are dominated by bunnies, by jackrabbits and cottontails, also sometimes termed lagomorphs, which is the fancy term. It's a very bad habit of mine that I use the Linnaean names all the time. So if I do that, I apologize. I try to explain when I can. Okay, so that's what we're talking about here in terms of just the basic taxonomic abundance snapshot right now. But one of the things that zooarchaeologists love doing is calculating these things called indices, index values. And so I'm going to go through some of the most common ones, and I'm going to explain them right now. I see some stuff popping up. If there's anything, just let me know. You can feel free to interrupt me. So the lagomorph index, just go off on this for a second. It is a ratio of cottontails to, right here, over jackrabbits plus cottontails. So basically, if this value, this index value, is closer to one, you have an assemblage, faunal assemblage, dominated by cottontails. If it's not close to one, then you have more jackrabbits. That's the idea here. So you're trying to get at understanding body size selection is one of the major things with these indices. So how does Haynie stack up against other sites in the Crow Canyon database? Well, actually, it has one of the lowest lagomorph index values, meaning there's more jackrabbits in the Crow Canyon database. This site right here, Sama, Pueblo, is actually a P4 site located in the northern Rio Grande. So this is not a Central Mesa Verde site, but it is in our database. Steve Wolverton analyzed this fauna. So it's in our database, but the rest of these are Central Mesa Verde, large Central Mesa Verde faunal assemblages, and Haynie has the lowest lagomorph index. Okay, so meaning more jackrabbits, not a lot of cottontails. Goodman Point, absolutely dominated by cottontails, okay? Not very many jackrabbits there at all. Smaller bodied animals were selected there. Okay, the artiodactyl index. Haynie has one of the largest artiodactyl indexes. And basically all that is a ratio, again, of like things like mule deer or pronghorn or bighorn, okay, over the mule deer, pronghorn, bighorn, and lagomorphs, cottontails, and jackrabbits. Again, body size selection. The closer to one that you are, the more deer and big game you're going after. So of the Central Mesa Verde sites, right now, Haney has the highest artiodactyl index value. Samo Pueblo, again, is that P4 later Northern Rio Grande site. But so I would say when you're looking at index values just in general, like if you were going to look like across North America, not only in the Southwest, this is still generally, I think people would consider this pretty low, but relatively speaking, it's one of the highest. Okay. The Turkey index is a little bit different. It has basically, this is a gauge of like focusing on domestication versus hunting. So we have turkeys that are one of the domesticated animals that Pueblo people domesticated in the past independently. Turkeys over jackrabbits, cottontails, and turkeys. Again, closer to one you are, the more like turkeys you're focusing on. Right now, of the Crow Canyon sites, Haynie has the lowest turkey index value. There's not a lot of turkeys that I've looked at. Now, that being said, which is a really interesting pattern, especially if we're thinking about what people are up to at the Lakeview group, what resource specialization is kind of maybe like, how people are relating to each other. But I can almost guarantee you, I can guarantee you that there are more turkeys that I have not looked at yet from this site. So this is an inaccurate snapshot, but I just wanted to communicate where we're starting out at. So those are the basics. Okay, basic indexes and things like that. One of the also really, really important things that I just have to talk about, right, is identifiability, right? I told you when we look at bones, bone fragments, we have to figure out what bone it is first, and then we can maybe talk about what kind of animal it is. If we can't figure out what bone this thing is, this specimen we're looking at, it becomes unidentifiable. It gets a code called UNI, unidentifiable. One of the things that's happening here at Haney, and I don't think I'm going to really be able to get to sussing this out today in this talk, but you can see it in the reports. I really dive deep into this topic. One of the things is that Haney has the lowest identifiability of the Crow Canyon sites that we've looked at across our database. Sama has the highest, Sand Canyon, next highest. Things are very identifiable. At Haney, things are not as identifiable, okay? So this basically means, so we have less than 50% identifiability right now. There are a lot of bone fragments that are very broken and don't have distinguishable diagnostic ends where I can identify them. So this is one of the most important questions surrounding Haney right now. And this is what requires a lot of data analysis. So that's maybe why I'm not going to go too deep into it. But if you want to know more, again, I've worked on this and we can talk about it more at the end of the talk. Okay, so that's about a lot of numbers and graphs. One of the things I want to talk about, it's really important and we've worked on, this is not just me, I also want to reiterate this. So Dave Satterwhite, Steve Copeland, Reuven Sininsky, Susan Ryan, we've all worked on these bison remains that have come from Haney. And we presented on this topic a poster at the SAAs last year, and bison are pretty rare in the Central Mesa Verde region. I know of, I think, 4 other specimens that have ever been identified from the Crow Canyon database, and then maybe 2 to 4 others that have been identified in the Central Mesa Verde region. We have over 6, and we just added another one, another toe, just a few weeks ago of bison from Haynie, which is absolutely fascinating. I have a little kind of schematic here of the things that we've identified from bison. So identified 2 bison to the species level. I've had humerus bits, lots of toe bits, a metapodial, and then a thoracic vertebra fragment, and then a rib fragment that I only got to say that it looks like a bison, like bison size. It was very chunky, but it just didn't have enough to really get it to enough morphology on the bone specimen itself to get it very far. So I was very hesitant to call that bison. Okay, so that is what we're dealing with when we're talking about bison. I took specimens in the center here I took specimens to the Museum of Southwestern Biology to double check that my IDs are right. There's tons of guides on this. But here is the comparative specimen, the modern bison specimen from the museum. And then here's an elk, which is the other thing this could be confused with, another very large artiodactyl hoofed mammal, even-toed hoofed mammals, artiodactyl. And what we can see is this kind of major articular facet, the general shape. This facet here, this facet actually right here is super diagnostic. The shape of this gets us to bison, very large. We had things like toes. So here, just to further try to convince you, this is the archaeological specimen. This is an elk, and these are bison toes here of known bison. One of the major things, you can't really see it so well in this picture, that sets these apart as a diagnostic feature, are that these bison toes have these like dimples, or kind of raised bits rather, not dimples, raised bits, like local raised bits here. And we also have that on these. Can't see it so well, but they exist, I promise. So again, bison toes. So of all the major, of all the skeletal elements that we're dealing with, that's what we have right here. They're highlighted. One of the major questions that came up from this is that we actually have these bison specimens all the way through almost like 2 meters, I think, worth of a vertical sequence, different stratigraphic sequence. We have multiple bison specimens, right? Maybe far apart through time. Maybe we have different individual bison. So it's rare enough to get one bison in the Central Mesa Verde region. But what if people at Haynie were actually getting bison regularly through time? So we started talking about, man, what if there's, multiple bison here? It could be so interesting. This is so fascinating. And then I had to put on my very serious zooarchaeology hat. And I had to say, when we do all the fancy zooarchaeology calculations, we think about bone overlap, we have what's called a minimum number of individuals, MNI, which is literally the count of the lowest amount of individuals this could be based off of bone overlap. We have an MNI of 1. Of all the bones that are represented within this sequence, you cannot make 2 skeletons out of it. There is no skeletal overlap is the point. We don't have, say, 2 right humeri or 2 right femurs or whatever, right? To say that, oh, this is definitely 2 individuals. So I had to say, maybe we need to test if this is indeed multiple individuals or 1. And so one of the ways that we devised to do this, this is not done yet, so that's also really important to point out. One of the things is we wanted to radiocarbon date to better understand this sequence, and say, okay, if we do calibrated radiocarbon dates and they're really far apart from each other, significantly far apart, then maybe we can say we've got multiple individuals. So we ran radiocarbon dates, AMS dates, on these bison bits. These are the PD numbers, the locations, and then the actual skeletal element itself next to each one. And I present them here like this, these calibrated dates. I present them here. This is the median date, and then the range, the 95.5% like error range for each one. And then this is actually how it calibrates on the curve, if I won't get too far into this. But basically, the punchline here is there's actually significant overlap here. We cannot use radiocarbon dating to say that there are different individuals. There could still be different individuals. They could be, because these are really broad date ranges. Okay, so we have like over 100-year span that these radiocarbon dates calibrate to, because they hit a certain part of the curve that wasn't in our favor. But we can't rule out that this still could be one specimen. One of the really, or one individual, I should say. One of the really telling things is this is the stratigraphic profile of this, and the scale is here, of this trench that these come from. And I've kind of tried to do this little like image to show where each one is located at in stratigraphic sequence here. And I'll kind of simplify this here in a second. One of the things that we can see, I'll kind of remove some of these and only focus on these three bones. They all have identical radiocarbon dates. They come from different portions. This first phalanx right here comes from a feature that is a subfloor feature, subfloor to this. This is what it looks like on the surface that was dug in here. So basically, the top thing from the bones that we have and the bottom thing date to the same time. So right now, one of the things that we're working on, and you got to stay tuned because we're going to keep working on this problem. I say we're chasing these bones through time and space right now. One of the things that we're working on is developing higher resolution Bayesian chronological models with radiocarbon dates, with annuals throughout the sequence. And Ruben Sininsky, our lab manager here at Crow Canyon, he is the one who is really, really good with this stuff. And we're working together to model like how long, how much time could this whole sequence have taken to have been deposited, right? Could it have been really rapid? Here in the Southwest, we look at something that's this big stratigraphically, and we think it has to be a really long time, hundreds of years. But some of the initial models that we've done suggest that this could be under 40 years. This whole sequence could be under 40 years. And this very well could be one individual. So that's a lot about bison. I'm going to breeze through this because I want to get to the wolf, and then we'll stop. One of the other things that we're really interested in is stable isotope values. And we ran stable isotopes on these bones. When you're doing AMS, they're paired, they're actually separated off and run. One of the things that you can do with carbon is you can actually place, so we've actually, from the bones, the chemical signatures, you can tell what these animals were eating based off of carbon. What kind of plants were these eating? What kind of grass for bison? What type of grass? And we actually have maps of grass types and how they plot in carbon space, carbon isotope space, and we call these isoscapes, and they've been developed. They're really cool. But basically what we can do is take these bones and try to plug them in on the landscape. And I need to do way more work on this. The question that we can answer with this is, were these bison somewhat locally procured, maybe coming from around the Four Corners region, generally speaking, or are they coming from the plains? Right now, how these plot out, just really briefly, how these plot out, they could very well, they're kind of plot out in this kind of pinkish area. They could plot in, or pink to orange, depends. They could be somewhat local. They're definitely not coming from the heart of the plains. They could be coming from some of the transitional zones, but I'm going to do more work on this in the future. And we're going to get some really much better maps on this. So that's what's going on with bison. I'm going to spend 4 minutes on this wolf specimen. Again, another really rare specimen to find in the Central Mesa Verde region. I know of 2 from the Crow Canyon database, 2 wolf specimens recovered, and then from the Central Mesa Verde, maybe region just in general, maybe another 2 that are securely identified to wolf, gray wolf. Well, we recovered a specimen that was a complete mandible from Haynie. This is what it looks like, a complete tooth row. It's absolutely fascinating. It's crazy to have this kind of specimen. This is the comparative specimen from the Museum of Southwestern Biology. I'm just going to give away some things here. Having the complete teeth allows us to age this wolf. We can maybe talk about the time, how old it was when it died. We call that ontogenetic age. And then we can also measure these teeth to help us say how confident can we be in this prediction of this is actually a wolf, and not say a really, really big domesticated dog or a really, really big coyote on steroids or something like that. So we're going to get into all that. I'm going to go really fast through it. I also just want to point out, this has got a really interesting kind of pathology associated with it, a really intense area of wear. This cusp right here, that's right here on this individual, is completely gone. I've talked with some much better zooarchaeologists than myself, and they said, I wouldn't make such a big deal about this just yet because it could be individual bite variation that's causing that. That's just a really intense area of wear. But one of the things that I did to help identify, to say how confident can we be in this, is I measured all these different areas here, the tooth row essentially in different ways, and had all a bunch of different values associated with that. And I plugged them in. I developed a model from a database that Martin Welker at the Arizona State Museum created, this huge database of North American canids of the same measurements. And then what I did is I created some predictive models, like you can think fancy machine learning models, to say how confident can we be in separating wolves from coyotes, from foxes, from domesticated dogs. So the reference specimens are all here in blue, red, purple, and then it's kind of hard to tell domesticated dogs apart, but these models are like beyond 95% usually accurate, just to begin with. So it's a really good model for prediction. And then this wolf specimen here that we're talking about plots over in this space. This is the archaeological specimen, the highlighted one. This is where it's at. When we ask the model, how uncertain are you about this identification? What it says is it is 100% certain of a 95% certain model. It's about as good as we can get for a taxonomic identification using this many comparative specimens. This is about as exhaustive as I can think of as a way to identify this mandible to wolf. And I have a huge debt to Martin Welker for creating such an amazing database that I could do this work with. Okay, the last thing, the last minute here, I wanted to know how old this individual was when it died, but I couldn't find data on when do wolves normally die in the wild. And so you think this would be a very easy thing to figure out. It's not. It's very biased by zoo data, right? Wolves that are being cared for. So I reached out to the Yellowstone National Park Wolf Project, the Yellowstone Wolf Project, and asked if I could use their data on this topic. And initially they were like, "Who are you?" And then I told them about this project, and they were like, "Oh, that's absolutely fascinating." So what we can see is total wolves normally die at the age of 3 years old or so. Right here, they peak here. Okay, and then it kind of goes off from there, their populations go. And when we split this up between males and females, it's about the same. Our wolf, the Haney wolf specimen, lived to an extraordinarily old age relative to a gray wolf, around 7 to 12 years old, based off some of those wear patterns, some of those really heavy wear patterns. Probably more between 7 to 10 years old is what that wolf was. But that locus of really hard wear made me kind of bump out this range. So we are talking about a wolf that could have been potentially cared for in some way or another. That is very speculative at this point, but it's possible that it made it into this later age class. And so I'm going to stop things there. I'm going to make some time for some questions. I told you I was going to talk ad nauseum. So I hope that was interesting. Okay.
That was fantastic. And you did not talk ad nauseum. So thank you. I do have, before I jump into the questions, I do have an answer from Dr. Ryan on the how common are half moon kivas. She says they are not common. They show up in Chaco great houses as well as in D-shaped structures in late P3 sites like Goodman Point and Sand Canyon.
Boom.
So there you go.
Thank you to Susan.
All right. Well, let's go ahead and jump into the question in the chat. Is there any way to use DNA testing with the bison bones?
Okay, so that is the really big question, and I'm hoping that is where we go with this. I wanted to use radiocarbon dating to begin with, though it is imperfect for this question, especially where we hit on the curve. That's in retrospect. Didn't know that when we began. But the aDNA would be the way to separate individuals, but it's more expensive, right? So I kind of wanted to do due diligence before we started using these more expensive methods. And just personally speaking, I've always wanted to learn aDNA techniques and haven't yet. So this would be a perfect project to kind of do that with. I do want to tell an anecdote, though, about radiocarbon dating these bison bones. So, you know, I took these bison bones and I'm IDing them as bison. I went to Albuquerque, spent time with the specimens, but there was this creeping thing in my head the entire time that was going, you know, bison are really hard to tell apart from domesticated cattle, like from, you know, skeletally speaking, it's some postcranially, it can be impossible to tell them apart. So the kind of punchline here is it's possible these are intrusive cows that somehow got into all of this and we just missed it. And I messed up the ID, right? And so I have to say that when these dates came back to where they should be if it was a bison, like where there's no cattle, domesticated cattle, I was relieved. I was like, I can do my job. So Yeah.
Well done. Well done. I had no doubts.
I
So
did.
one of don't admit that. Yeah. One of our friends, this is kind of a fun question. Are there any feline bones at Haney?
Yeah, actually there is. There is a really interesting specimen, a bobcat terminal phalanx, so like the fingertip, the claw tip, bobcat terminal phalanx that was drilled out, like it was part of some kind of like a pendant of some kind or some kind of jewelry, like it was strung through something. And it actually looked like the drill like broke through the side. It could have been preservation. I couldn't really tell, but yeah, we do have bobcat specimens. I have identified at other sites, not at Haney, mountain lion specimens. So those are we're always on the lookout for those too, and they're definitely around these parts. So yeah,
Yep. That's really cool. Yes, we have a mountain lion in our backyard.
yeah.
Yes. Someone asks, what evidence on the bone itself do you have regarding methods of preparation and eating? Uh,
Ah, yeah. So for the bison, if we want to be specific about that, none. No cut marks that I can find. They are broken. Some of them are broken in what we call a spiral fracture, which some people say is indicative of like marrow use. So you could maybe argue that is indicative of like
mhm,
that behavior, getting to within bone nutrients. But on other bones, just in general in the Haney fauna, we get all kinds of stuff. We get the cut marks, we get burning. Burning is usually the one that it's the kind of the smoking gun for cooking. And they say that the bones, when they're being roasted, things are like bone is exposed and the meat is being roasted, that you'll actually see localized burning on the exposed part of the bone. Like it's just very like black or white on one small part of it. And so we have evidence of that too. Yeah.
excellent. So a question about kind of time periods, right? Are Haney and the comparison sites all the same time period? Um, or are they later? They're basically asking if the difference could be accounted for by a time effect, like increase or decrease in hunting and population.
Absolutely. That could be what we're talking about. There's like in terms of the comparisons that I made with the basics data, some of those sites are later in time, and absolutely there could be things that are on the landscape at that time. So that could be something. In terms of the Lakeview group, that is a really interesting question. I do not personally have a handle so much on um, the other great houses, Ida, Jean, and Wallace, in terms of like earlier components and like getting those fauna out analytically and looking at them and comparing to Haney, but that is definitely on my agenda. It's an important thing if we're talking about the development of these kind of great house clusters. I don't know if I'm going to be able to do it from what was reported, but I'm going to try. I'm going to really, really try.
Someone is curious about things designated as having low identifiability. Is that about processing or cooking style, or do we know?
Yeah, so I'll just give give it away. So what I've been working on is using the last the two reports, and I'll return to this topic in this next report. It's working with these fancy machine learning models, these AI models, to say, given the taphonomic data that I have recorded, given those as like predictor values, can you identify or can you predict unidentifiability? So what combination, what suite of these different things can you tell me that leads to unidentifiability at the Haney site? And I've developed some models that basically tell me that unidentifiability is strongly associated with, at Haney, shaft fragments and thick shaft fragments, so from medium artiodactyls. Usually when I record these shaft fragments, so like I think the long bone, this right here, I will record just like a category that says like thick cortical bone or something like that. And so what I'm thinking right now, this could change. So don't quote me on this, but what I'm thinking is it actually could have been processing of bone by Haney occupants that caused this unidentifiability issue. I've looked at there's areas of disturbance, major areas of disturbance at this site, and I've done other analyses with those to rule those areas out to say that the disturbance zones aren't driving unidentifiability. So if you look at the reports, you'll see that. And again, this could change with more data. We're over only like a quarter of the way through the analysis. So
Very cool. Thank you. So were the dogs in people's diet?
yeah, there's evidence for that. And I would just caution people to think outside of your culture, to think more relatively about culture. Why wouldn't dogs be a part of the diet, right? So I will say, though, the zooarchaeological evidence for that in the Southwest is not extensive. The one area that I know of where it has been argued for convincingly is Arroyo Hondo, a Pueblo 4 site around Santa Fe or in the northern Rio Grande. I don't know of any evidence for it for domesticated dogs being consumed in the Central Mesa Verde region. Not saying it doesn't exist, but it's certainly a possibility.
So one of your favorite questions, or not, I'm actually not sure if you like this or not. Can AI be used to identify your shapes and sizes of animal bone fragments?
Yeah, that's what I've done. That's
That's
exactly
what
what
you've
I
done.
Yeah. So those, when I say machine learning models, that's the learning part, the intelligence part. They get conflated, and the machine learning thing is just a fancy buzzword for specific techniques and ways to split up data to basically learn a model to do specific tasks. One of the things that I'll get on my soapbox about really briefly is that machine learning and AI is much more mundane than you probably think it is. Most people think it is. I'll like qualify that. People usually want to talk about like Terminator, and when I want to talk about AI, I'm talking about data, and people are like, "Oh, that's boring," and I'm like, "But that's what we're talking about." So, yes, the short answer, yes, and that was my little diatribe about AI.
Yes, we remember the AI is not as sexy as you think it is
Yes.
a Jonathan Dombrosky quote.
Yeah.
I
Yep.
think this will be our last but not least question. Kind of a fun one. Can DNA analysis be done on the bone of a single species to determine number of individuals?
Oh, what a great question. Okay, so I'm going to kick it to, and that's a really, whoever asked that question, awesome. I'm going to kick it to some work that Steve Wolverton, a fantastic zooarchaeologist, has done with colleagues in Argentina, where they've tried to look at effective population sizes from genetic diversity on guanacos, which are camelids, wild camelids down there in South America and Argentina. So the short answer is theoretically, yes, you can get an effective population size from what we know today, some error bars. The error bars can be pretty big, but ADNA is definitely something that needs to be further explored. And I've thought about it so much, and it's just fascinating. But I'll leave it there. So possibly, theoretically, yes, there's some methodological hurdles, but yeah.
Very cool. Possibly somewhat expensive is my
Yes.
managerial brain. Like, oh my God, that would cost a fortune.
Yeah.
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 Well, thank you so much, Jonathan. And thank you to everyone for watching. What a spectacular presentation. And thank you to everybody for supporting Crow Canyon. And we hope to see you next week.
Thanks, everyone. Bye.